So in the last class, Bhairesh had asked me that he had a doubt regarding the recombinant DNA technology, which we are going to discuss regarding the concept. Later on, we are going to discuss regarding multiple choice question, right? So let us begin. So here, I actually wanted to use board for this recombinant DNA technology. Due to my health issue, I'm unable to stand for long hours and explain. So that is why I used PPT actually. Okay, so can you able to see the screen? Just to tell me.
Yes, ma'am.
Yeah. So in introduction part, a recombinant DNA technology. What do you mean by recombinant? Re means what? Repeat. Combinate means what? Combination. So here we are combining. Here we are combining the DNA of two organisms where we want a one character or one certain profit from it. So for example, the DNA structure in every organism, you take animal, you take plant, you take human beings, the DNA structure will be the same. Means it is made up of nucleotides, adenine, guanine, cytosine and thymine. So that will be same. Okay. So the length of a DNA, that all may differ, but the composition of the DNA will be same. Yes, who's this? Can you mute yourself, please? Yeah. Fine. So the component of the DNA will be the same. So here, what this recombinant DNA technology means, it is a laboratory method where genetic material of an organism is collected and it is multiplied in other organism, so that when that multiplied DNA, okay, it will be inserted into another organism. ಆ ಮಲ್ಟಿಪ್ಲೈಡ್ ಡಿ ಎನ್ ಏನಿರುತ್ತೆ, ಒಂದು ಆರ್ಗ್ಯಾನಿಸಮ್ ಇಂದ ಒಂದು ಡಿ ಎನ್ ಎನ ಐಸೋಲೇಟ್ ಮಾಡೋದು, ಸಪರೇಟ್ ಮಾಡೋದು. ಓಕೆ, ಬಯಾಲಜಿಕಲ್ ಹೇಗೆ ಸಪರೇಟ್ ಮಾಡ್ತಾರೆ ಅದನ್ನೆಲ್ಲ ಆಮೇಲೆ ತಿಳ್ಕೊಳ್ಳೋಣ. ದಟ್ ಇಸ್ ಇನ್ ನೆಕ್ಸ್ಟ್ ಸ್ಟೆಪ್. ಸೊ ಬೇಸಿಕಲಿ ಏನ್ ಮಾಡ್ತಾರೆ ಅವರು, ದ ಫಸ್ಟ್ ಒನ್ ಇಸ್ ಐಸೋಲೇಷನ್ ಅಂತ ಮಾಡ್ತಾರೆ. ಒಂದು ಆರ್ಗ್ಯಾನಿಸಮ್ ಇಂದ ಆ ಡಿ ಎನ್ ಎನ ತಕ್ಕೊಂಡು ಅದೇನ್ ಮಾಡ್ತಾರೆ, ಆ ಡಿ ಎನ್ ಎನ ಮಲ್ಟಿಪ್ಲೈ ಮಾಡ್ತಾರೆ ಇನ್ನೊಂದು ಆರ್ಗ್ಯಾನಿಸಮ್ ಅಲ್ಲಿ. ಸೊ ಆ ಮಲ್ಟಿಪ್ಲೈ ಆದ ಫ್ರಾಗ್ಮೆಂಟ್ಸ್ ಅನ್ನ ಏನ್ ಮಾಡ್ತಾರೆ, ನ್ಯೂ ಆರ್ಗ್ಯಾನಿಸಮ್ ಅಲ್ಲಿ ಇನ್ಸರ್ಟ್ ಮಾಡಿದಾಗ ನಮಗೆ ಬೇಕಾಗಿರೋ ಕ್ಯಾರೆಕ್ಟರ್, ನಮಗೆ ಬೇಕಾಗಿರೋ ಪ್ರೋಟೀನ್, ಯಾವುದೋ ಒಂದು ಸಬ್ಸ್ಟೆನ್ಸ್, ಏನು ಯಾಕೆ ನಾವು ಅದನ್ನ ಸಪರೇಟ್ ಮಾಡಿದೀವಿ, ಆ ರೀಸನ್ ಏನಾಗುತ್ತೆ ಅಲ್ಲಿ ಫುಲ್ಫಿಲ್ ಆಗತ್ತೆ. ಓಕೆ, ಸೊ ಹಿಯರ್ it is formed by a laboratory methods of genetic recombination. So it is also called as molecular cloning. ಯಾಕೆ ಮಾಡ್ತಾರೆ, to creating sequence that would otherwise be found in the genome, to creating the sequence which is not found yet now. Okay, then rDNA is possible because DNA molecules from all organisms share the same chemical structure. As I said, the chemical structure of all the DNA molecules is same. That is why we can combine the DNA. They differ only in the nucleotide sequence within that identical overall structure. Nucleotide sequence will be changed. That is, first we can say adenine is binded with thymine. So in the other organism, there the first guanine is binded with cytosine. So the nucleotide sequence, the arrangement of nucleotides will be different. Pairing will be same. The arrangement, ಒಂದರಲ್ಲಿ ಅಡಿನೈನ್ ಫಸ್ಟ್ ಇದ್ರೆ ಇನ್ನೊಂದರಲ್ಲಿ ಗ್ವಾನೈನ್ ಫಸ್ಟ್ ಇರುತ್ತೆ, ಒಂದರಲ್ಲಿ ಗ್ವಾನೈನ್ ಫಸ್ಟ್ ಇದ್ರೆ ಇನ್ನೊಂದರಲ್ಲಿ ಅಡಿನೈನ್ ಫಸ್ಟ್ ಇರುತ್ತೆ. ಸೊ ಲೈಕ್ ದಟ್. ಫೈನ್. ಏನ್ ಮಾಡ್ತಾರೆ ಆಕ್ಚುಲಿ ಆರ್ ಡಿ ಎನ್ ಎ, ದಟ್ ಇಸ್ ರೀಕಾಂಬಿನೆಂಟ್ ಡಿ ಎನ್ ಎ ಟೆಕ್ನಾಲಜಿಯಲ್ಲಿ ಏನ್ ಮಾಡ್ತಾರೆ, it is a general name for a piece of DNA that has been created by the combination of at least two strands. It is nothing but a small DNA fragment only. How it is formed, ಹೇಗೆ ಮಾಡಿದ್ದಾರೆ ಅದನ್ನ, combination of two strands, combination, combining two strands of DNA. So DNA molecules are called as chimeric or chimeric DNA. So the pronunciation differs from region to region. So here chimeric means what? In Greek ದೇಶದಲ್ಲಿ, ಗ್ರೀಸ್ ದೇಶದಲ್ಲಿ ಏನಾಗಿದೆ, ಒಂದು ಮಿಥಿಕಲ್ ಅಂದ್ರೆ ಒಂದು ಪುರಾಣದಲ್ಲಿ ಒಂಥರ ಒಂದು ಆನಿಮಲ್ ಬರುತ್ತೆ. ಆ ಆನಿಮಲ್ ಹೇಗಿದೆ ಅಂದ್ರೆ, ದಟ್ ಈಸ್ ಅ ಫೀಮೇಲ್ ಆನಿಮಲ್ ವಿಚ್ ಹ್ಯಾಸ್ ಅ ಲಯನ್ ಕೈಂಡ್ ಆಫ್ ಅ ಹೆಡ್ ಅಂಡ್ ವಿಚ್ ಬ್ರೀತ್ಸ್ ಫೈರ್, not air, which breathes fire. It is a mythological character or a ಪುರಾಣದಲ್ಲಿ ಒಂದು ಕ್ಯಾರೆಕ್ಟರ್ ಗ್ರೀಕ್ ಅಲ್ಲಿ, ಹೌದಾ? ಸೊ ಇಲ್ಲಿ ಕಾಂಬಿನೇಷನ್ ಏನಿದೆ, ಲಯನ್ ಹೆಡ್ ಇದೆ, ಬ್ರೀದಿಂಗ್ ಆಫ್ ಫೈರ್ ಇದೆ, So like that, our DNA ಏನು ಅದಕ್ಕೆ ಹೋಲಿಸ್ತಾರೆ chimeric DNA, because they can be made of material from two different species. ಸೊ ಅಲ್ಲಿ ಮೈಥಾಲಜಿ, ಆ ಪುರಾಣದಲ್ಲಿ ಆ ಆನಿಮಲ್ ಏನ್ ಬರುತ್ತಲ್ಲ, ದಟ್ ಈಸ್ ಅ ಫೀಮೇಲ್ ಆನಿಮಲ್, ಅದು ಎರಡು ಸ್ಪೀಶೀಸ್ ಅಥವಾ ಎರಡು ಆನಿಮಲ್ ಕ್ಯಾರೆಕ್ಟರ್ ನ ಒಳಗೊಂಡಿದೆ ಅಂತ ಹೇಳ್ಬೋದು. ಫಾರ್ ಎಕ್ಸಾಂಪಲ್, ವಿ ಕ್ಯಾನ್ ಟೇಕ್ ಇನ್ ಅವರ್ ಮೈಥಾಲಜಿ ರಿಗಾರ್ಡಿಂಗ್, ನಾವು ನಮ್ಮ ಮೈಥಾಲಜಿಯಲ್ಲಿ ನರಸಿಂಹ ದೇವ್ರನ್ನ ತಗೋಬಹುದು. ಹೌದಾ? ಸೊ ನರಸಿಂಹ ಅಂತ ಯಾಕಂತಾರೆ? ನರ ಮೀನ್ಸ್ ಮನುಷ್ಯ, ಸಿಂಹ ಮೀನ್ಸ್ ವಾಟ್? ಲಯನ್. ಸೊ ಹಿಯರ್ ದೇರ್ ಇಸ್ ಅ ಕಾಂಬಿನೇಷನ್ ಆಫ್ ಟೂ ಸ್ಪೀಶೀಸ್. So that is called as chimeric DNA. See here the combination of a DNA. Okay. So that is rDNA or recombinant DNA. So rDNA technology uses palindromic sequence and leads to the production of sticky and blunt ends. rDNA ಅಥವಾ recombinant DNA ಏನ್ ಫಾರ್ಮ್ ಆಗಿರುತ್ತಲ್ಲ, so how it will end? Based on the palindromic sequence. What do you mean by palindromic sequence? For example, Gadag, G-A-D-A-G, Gadag. So this is a palindromic. When you reverse it also, you will get the same spelling. So like that, nucleotides also will be having the reverse. Suppose A-T-T-G, one strand alidre, innond strand aliratte G-T-T-A. So ನೀವು reverse ಮಾಡಿದಾಗ same spellings ಅಥವಾ same nucleotide sequence ಬರ್ತಿರತ್ತೆ. So that is called as palindromic sequence. All of you understood regarding the introduction? Please let me know in the chat box or through a mic.
Yes, ma'am.
Yeah.
Yes, ma'am.
Okay. Then, so first, what is recombinant DNA technology? So this is an artificial DNA. It is a combination of artificial DNA. Idu natural ಆಗಿ ಕಾಣಿಸಿಗೋದಿಲ್ಲ. This is an artificial method or a laboratory method. So what we are mixing? We are mixing two DNA from the different organism. So that is the simple definition of recombinant DNA. Repeat. Okay. Then combination DNA. Okay. Now. Then recombinant DNA technology ಮಾಡೋದ್ರಿಂದ ನಮಗೆ ಏನು ಯೂಸ್ ಇದೆ? We can isolate and clone single copy of a gene or a DNA segment into an indefinite number of copies which are all identical. The first and foremost importance is, for example, let us consider a DNA fragment. So our DNA fragment alli, we know that the DNAs are made up of a structural and functional units called gene. So one gene, one character ige ಏನಾಗಿರುತ್ತೆ? Responsible ಆಗಿರುತ್ತೆ. So if we want that gene which is responsible, let us take a gene which is responsible for the intelligence. Okay. So that gene we want. So instead of taking all the DNA, ಎಲ್ಲ organism ಅಲ್ಲಿರೋ ಎಲ್ಲ DNA ನ ಕಲೆಕ್ಟ್ ಮಾಡ್ಕೊಳ್ಳೋ ಬದಲು ನಾವೇನ್ ಮಾಡ್ಬೇಕು? ಆ ಜೀನ್ ಇರೋ ಫ್ರಾಗ್ಮೆಂಟ್, ಆ ಜೀನ್ ಇರೋ ಯಾವ ಪ್ಲೇಸ್ ಅಲ್ಲಿ, ಯಾವ ಪ್ಲೇಸ್ ಆಫ್ DNA ಅಲ್ಲಿ ಆ ಜೀನ್ ಇದೆ ಅಲ್ಲ, so that fragment we should isolate. So that will be happen during recombinant DNA technology. So ಈ ಟೆಕ್ನಾಲಜಿಯಲ್ಲಿ ಆ ಫ್ರಾಗ್ಮೆಂಟ್ ಅನ್ನ ಅಷ್ಟೇ ಐಸೋಲೇಟ್ ಮಾಡೋ ಮೆಥಡ್ ಇದೆ ಅಂತ ಹೇಳ್ತಾರೆ. Then when you isolate, you can multiply it into indefinite number. ಒಂದಿಷ್ಟೇ 10 ಕಾಪಿ 20 ಕಾಪಿ ಆಗತ್ತೆ ಅಂತಲ್ಲ. So we can multiply it into many or indefinite number of copies which are all identical. Identical ಅಂದ್ರೆ same. These new combinations of DNA, genetic material or recombinant DNA molecules are introduced into the host cell where they propagate and multiply. The technique or methodology is called recombinant DNA technology. So all these copies or DNA fragments, what they are done? They are introduced into the other organism or other species. So our species alli enagutte, these DNA fragments will multiply. Okay, will become more. So this introduction of new DNA fragments from one organism to another organism is called as recombinant DNA technology. I hope the summary is clear for everyone. So overall recombinant DNA technology ಏನಾಗುತ್ತೆ ಅನ್ನೋದು ಕ್ಲಿಯರ್ ಆಯ್ತು. ಕ್ಲಿಯರ್ ಆಯ್ತಾ? Yes. Hello. Is it clear?
Yes, ma'am.
What about others? Only Bhairesh asked that doubt. Only he is saying this. What about others?
No doubt, ma'am.
Yes, ma'am.
Okay, fine. So obtaining our DNA, how we will get the recombinant DNA? So we have several steps for that. Let us see the first step. The DNA fragment containing the gene sequence to be cloned, also known as insert, is isolated. So the DNA fragment which we want, which gene we want, so that DNA fragment, what it will do, it will be isolated, separated from the actual organism. So that gene which we will separate, no, it is called as GOI, that is gene of interest, gene of interest. The step 2 is cutting DNA. So what the fragment you took, that has to be cut and then inserted. Okay, the next one is cutting DNA. Step 3 is joining DNA. Step 4 is insertion of these DNA fragment into the host cell. So joining DNA, after joining DNA, what we are doing? We are introducing, we are inserting the DNA into a host cell through a vector. Vector means what? It acts as a vehicle which carries a DNA molecule which we want. Okay, so that vector will be usually the plasmid of a bacteria. Bacterial structurally, now kaltidivi, the bacteria will be having a specialized round DNA structure, circled DNA structure, which is called as plasmid. So those plasmids will act as a vectors, that is the vehicles which carry the desired DNA, the interested DNA. Okay, now, so let us study the steps one by one. So step 5 is rDNA molecules are generated when the vector self-replicates in the host cell. So what we did first, we took that DNA fragment and we put it in a vector. That vector we will introduce into the organism or a host. So in the host, plasmid will multiply. In the host, what happened? The plasmid will undergo replication. Self-replication means self-multiplication. Then transfer of the rDNA molecule into an appropriate cell. So after replication, self-replication, what happens? The transfer of DNA fragment from the vector into the organism. So if into which cell it has to be introduced. So that will be done. Step 7, selection of the host cell carrying rDNA molecule using a marker. So what happened? The host cells which will be having rDNA. So ಎಲ್ಲ cells ಅಲ್ಲೂ plasmid release ಮಾಡಿದೆ DNA ನ ಹಾಗಿರಲ್ಲ. So some of the cells will be having the rDNA molecule. Some of the cells may not be having. So we have to select the host cells which carry the DNA, that is rDNA molecule. So using a marker, we using a biological marker, not a marker which we will write on the board. Using that, we will select the cells. Then step 8, replication of the cells carrying rDNA molecule to get a genetically identical cells or a clone. So whatever host cells we collected, which has our gene of interest, our DNA fragment, okay, so that will be, and that will be allowed for multiplication. That will be allowed for multiplication to get many cells or to get many copies. Clone means the identical copy. Then, so let us study regarding the first step in detail. Okay, so the first step in making recombinant DNA is to isolate donor and vector DNA. Donor means from which organism we are taking the DNA. So that is a donor DNA, which donates its DNA. Isolate donor and vector. Vector means which plasmid. We have to isolate the plasmid from the organism that is like bacteria. Okay, the procedure used for obtaining vector DNA depends on the nature of the vector. So vector DNA, ಈಗ ಬ್ಯಾಕ್ಟೀರಿಯಾ ತಗೊಂಡಾಗ ಬ್ಯಾಕ್ಟೀರಿಯಾದಲ್ಲಿ ಪ್ಲಾಸ್ಮಿಡ್ ಆಕ್ಟ್ಸ್ ಆಸ್ ಅ ವೆಕ್ಟರ್, ದಟ್ ಇಸ್ ಅ ಸರ್ಕ್ಯುಲರ್ DNA, ಎಕ್ಸ್ಟ್ರಾ ಸರ್ಕ್ಯುಲರ್ DNA. ಸೊ ಆ DNA ನ ನಾವು ಬ್ಯಾಕ್ಟೀರಿಯಾದಿಂದ ಸಪರೇಟ್ ಮಾಡಬೇಕಾದ್ರೆ ವಿ ಹ್ಯಾವ್ ಡಿಫ್ರೆಂಟ್ ಮೆಥಡ್ಸ್, ಡಿಫ್ರೆಂಟ್ ಪ್ರೊಸೀಜರ್. ಎಲ್ಲಾ ಬ್ಯಾಕ್ಟೀರಿಯಾ ಒಂದೇ ಮೆಥಡ್ ಇಂದ ನಾವು ಸಪರೇಟ್ ಮಾಡ್ತೀವಿ ಅಂತ ಅಲ್ಲ. ವಿ ಹ್ಯಾವ್ ಸರ್ಟನ್ ಮೆಥಡ್ ಲೈಕ್ ಅಲ್ಟ್ರಾ ಸೆಂಟ್ರಿಫ್ಯೂಗೇಷನ್, we have another chemical methods. So based on the nature of the vector, based on the nature of a plasmid in which bacteria it is present, so we will select the procedure. So bacterial plasmids are commonly used vector. As I said, bacterial plasmid commonly used vector or vehicles. These plasmids must be purified away from the bacterial genomics DNA. So e plasmid ಏನಿರತ್ತಲ್ಲ ಬ್ಯಾಕ್ಟೀರಿಯಾದಲ್ಲಿ, so these are a extracircular DNA. So DNA of a bacteria will be different. So these plasmids are the extracircular DNA. Even though these are extracircular DNA, they will be having the genes which are related to bacteria. That is bacterial genomics genes. So that has to be removed so that we will get a purified plasmid. We will get a purified plasmid in which we are going to insert our gene of interest or our DNA fragment. Okay. So this is about isolation. All of you got regarding the step of isolation? Text me.
Yes, ma'am.
Okay. Fine. The next step. Now we inserted it. Cutting DNA. So what we have to make? The restriction enzymes. What are the restriction enzymes? The restriction enzymes are the enzymes or a biocatalyst which cut the exact nucleotide sequence or a DNA fragment how much ever we want. So the restriction enzyme of E. coli, we know that E. coli is a bacteria. E. coli is a bacteria. So its plasmid acts as a vector. The restriction enzymes E. coli cuts a circular DNA molecule. It cuts a circular DNA or a plasmid bearing one target sequence. Bearing one target means whichever target we want, resulting in a linear molecule with single stranded CK end. So here C. So this is a sequence of circular DNA. Only the restriction endonuclease cuts this part. Okay. So with single stranded, with a linear means the circular becomes straight now. Straight, after that the cutting occurs which becomes a straight, okay, which is a straight molecule. Then after that with the single stranded sticky end. Single stranded means single strand, not a double strand, and the end will be sticky. So that is a cutting DNA. See here. 1 minute. So this is E. coli. Okay. So this is a recognition sequence, means this sequence we want. Okay. So what we are doing? The restriction enzymes are acting on these nucleotides and they will cut. So the cleavage occur. The cleavage means the cut occurs. So this cut will be having the G and C will be separated. The G will be separated. So for that, this end will get sticky end. C. AATTC, then CTTAA, CTTAA, AATTC. So these will be having the sticky ends. These both will be having the sticky ends. Okay. Now, the next one is insertion, means inserting the DNA fragments. Choosing a gene cloning vector. First, what we have to do? We took the DNA fragment which we want now. So which nucleotide sequence or which gene of interest we want. So that DNA fragment we separated. Now we will insert or we will make the DNA fragment to sit in a vehicle. So for that we have to see which vehicle we have to select. Okay. A vector is any DNA molecule which is capable of multiplying inside the host to which our gene of interest is integrated for cloning. Very nice. See here. What is vector? It is a DNA molecule. So what it do? It has a capacity to multiply, to replicate inside the any other organism. So inside any other means that organism is called as host. How it will multiply? Along with the DNA which we have inserted. That is, host to which our gene of interest is integrated, means is mixed. For cloning means what? For copying. So whenever vector multiplies, along with the vector, our gene of interest is also multiplying, is getting replicated, is also getting copied. So in this process, what happens? Restriction enzymes function as scissors for cutting the DNA molecule. Now, so that those DNA fragments has to be cut and they have to be inserted into the vector. So directly you can't do or put the DNA molecule into the vector. So you have to attach it to its strand. So for that we want scissors. So we don't have the physical scissors there. So there the restriction enzymes acts as a scissors. Then ligase enzyme is the joining enzyme that joins the vector DNA with the gene of interest. So ligase enzyme, after cutting and inserting it into a circular DNA, so that DNA has to attach to that vector DNA. So that will be done by a fevicol like a substance or a glue like a substance, which is called as ligase enzyme. Okay, then so after its multiplication, that is a vector DNA along with the gene of interest, I am repeating, the vector DNA along with the gene of an interest or a DNA fragment is called as recombinant DNA. So that is called recombinant DNA. Now, so vector is ready with the gene of interest. So that vector or the plasmid of a vector has to be introduced into the host cell. So how it can be done? Plasmid vector, the vector is added to a flask containing a culture of E. coli. So first what we'll do, the vector is added or inserted into the flask, okay, into the conical flask, which contains a culture, which contains a colony of a E. coli. Culture means they are prepared in the lab itself. So calcium ions, usually in the form of calcium chloride, are added to the flask followed by a brief heat shock. So that conical flask will be heated. Then after that, calcium chloride is added. Okay. Why? Because they need a calcium ions. So for the calcium ions, calcium chloride has been added. What happens? This allows holes to briefly appear in the cell surface membrane of the E. coli, making it permeable to DNA and allowing the plasmids to enter. So the plasmids has to enter into the E. coli. So the cell surface of a E. coli, what happens? They should contain or they must get the holes. So these calcium ions will be involved in making the holes on the cell membrane of the surface of a E. coli, so that the plasmid will enter into the E. coli. Next, phage vectors. So what are these phage vector? Introduced by infection of bacterial lawn growing on an agar plate. So it is an infection, okay, infection. Then the culture or growth of virus. Once you introduce it in the culture may damage or that organism will only grow. That organism means the virus will only grow in the culture. Then now all happen, DNA molecule, plasmid DNA, placing the gene in the vector. So how they will place? DNA molecule will be small and separated based on the size, based on smaller, larger, based on that they will be separated. So bacterial cells are broken up and chromosomal DNA centrifuged down. So the bacterial cell or E. coli here, they will break down and the chromosomal DNA, whatever present, will be, shifted for centrifugation process. So centrifugation, what happens? This leaves the plasmid DNA in the liquid above the palate. So at the residue, at the bottom, we will get chromosomal DNA. And the upper part, we will get the plasmid DNA. Okay. Then the plasmid are purified before cutting with a restriction enzyme. Then, so restriction fragments will be there, no? The restriction or a restricted. Okay. From the donor DNA means from actually which organism we took the DNA. will be mixed with the plasmid DNA and joined by the sticky ends. The initial attraction is due to the hydrogen bonds. So how they will be joined? It is due to the hydrogen bond. But the main thing is sugar phosphate. Sugar we know, phosphate group we know, which forms the backbone of a DNA. Here also these will form the backbone of a DNA, and those will be joined using ligase enzyme only. Just a minute. Yeah. Next. So this is regarding recombinant DNA technology. Once they are introduced into the vectors and once they are introduced into the host cell will multiply and the plasmid will be collected by a centrifugation. From that we will get a desired DNA fragment. So I hope the steps are clear for everyone. Yes.
Yes, ma'am.
Okay. So here example of use of recombinant DNA technology. So where we will use recombinant DNA technology? Very important question. Yes. So the first one is insulin reproduction. Insulin. So what is insulin? It is a hormone which maintains glucose level in our body. So can anyone tell me where the insulin is produced in our body? Yes. Where the insulin is produced in our body? Yes.
Ma'am, pancreas.
Pancreas. What about others?
Yes, ma'am. Pancreas.
Pancreas. Okay. Janvi?
Ma'am, what is the question, ma'am?
Where in which part of our body insulin is produced?
Pancreas only, ma'am.
Mhm. So the pancreas are the glands which produces the insulin. Okay. Now, so here the DNA for insulin is first isolated. Why insulin reproduction occurs means we all know that now most of the people will be having the diabetes. Okay, so they will be in need of the insulin injections. So how that insulin is prepared, that we are going to do. The DNA for insulin means the gene which is coded or which is related to the insulin production will be separated. Then a plasmid made of DNA is removed from bacterial cell. So a plasmid will be removed from a bacterial cell. Then a restriction enzyme cuts the plasmid. So which enzyme cuts the plasmid? A restriction enzyme. So what happens? The plasmid DNA will open, leaving the sticky ends. So that when they will open, the ends will be sticky. So this insulin gene will be introduced into this into the plasmid. Then after introducing, DNA ligase enzyme joins the insulin gene with the plasmid DNA. Now, the plasmid is inserted back into the bacteria. Then what happens? The bacteria will be the host cell. The bacteria will multiply and produces a many copies of plasmid. So after that, a bacterium makes human insulin using the gene in the plasmid. So which insulin is produced? Human insulin is produced in the plasmid and the bacterium is undergoing the division so that the plasmid is also undergoing the replication. So after that, the plasmid is again separated from the bacteria. The plasmids are undergone centrifugation where we will get the insulin. The insulin is extracted from the bacterial culture by a method of centrifugation. See here. Yes. So this is a plasmid. This is a plasmid. Okay, vector. Vector means a plasmid of a bacteria. Vector means the vehicle carries foreign DNA molecule. Foreign DNA, the DNA from another organism. So this is a foreign DNA. So cleavage by E. coli endonuclease. E. coli means restriction endonuclease. Cleavage occurs. So here also cleavage by endonuclease. So the required or the suggested or the required sequence will be attached to this one, annealing. So this attachment is called as annealing. So DNA like this. See here the DNA fragments are not joined. So when the DNA ligase acts on the ends, it will be joined. So this is a plasmid chimera. Okay, hybrid plasmid we can call. Next, after that, okay, the plasmid is then introduced. See, DNA insertion occurs. This is a foreign DNA. Then by the E. coli, we will separate the strands, and the strands are introduced into the plasmid. So this is called as recombinant DNA. This recombinant DNA is again introduced into the bacterial cell where the multiplication occurs. Almost all the bacteria will get that rDNA molecules. After that, bacteria plated on the medium plus antibiotic. Antibiotics are added. Only bacteria containing okay. So when you put the antibiotic means which will kill the bacteria. Okay. So the cells, the bacterial cells which don't have rDNA, that is recombinant DNA, will be killed because those are the normal bacteria which will be killed by the antibiotic. Why the antibiotic will not kill the hybrid bacteria or the clones? Because now the bacteria which got a new plasmid, so it has a mixture of DNA, it has a different DNA. So that will not kill by the antibiotic. So the bacteria which don't have rDNA, those will be killed by the antibiotic. Then the remaining will be only having the bacteria which have the rDNA molecule. So those will be grown and those will be cultured and centrifuged. Then we will get the rDNA or recombinant DNA molecules. Okay, see, human cell insulin gene is isolated. So here bacterial cell plasmid is isolated. Now using the E. coli restriction enzyme. So what happens? Yeah, using that the plasmid is cleaved or joined with the insulin gene. Then again the plasmid is introduced into the bacteria. Now the bacteria is called transgenic, means the gene sequence has been changed. Trans means different, genic, gene. Okay, and these are grown in a culture, and then we will put antibiotic. The only normal bacteria will die, transgenic bacteria will survive. So from those transgenic bacteria we will extract the insulin. Okay, I hope the insulin production is clear for everyone. Yes? Okay. Find the applications and then. So we will just finish up this application and we will move to the discussion of question and multiple choice questions. So applications, preparation of gene maps of several organisms, we will use DNA, rDNA technology. Then in revealing details of various infections like inborn errors, ಹುಟ್ಟನೆ ತುಂಬಾ ಮಕ್ಕಳಿಗೆ ಏನೋ ಒಂದು ಎರರ್ಸ್ ಇರ್ತವೆ ಅಥವಾ ಪ್ರಾಬ್ಲಮ್ಸ್ ಇರ್ತವೆ, so that will be detected by rDNA. Finding out complete nucleotide sequence, one genome in a complete nucleotide sequence ನಮಗೆ ಬೇಕಂದ್ರೆ ನಮಗೆ rDNA ಟೆಕ್ನಾಲಜಿ ಇಂದ ಗೊತ್ತಾಗುತ್ತೆ. Detecting cytogenetic abnormalities, that is Down syndrome, atherosclerosis, coronary artery disease. So this all will be detected using this method indirectly, not directly. Preventing various genetic disorder, for example, inherited hemoglobin disorder, phenylketonuria, retinoblastoma. So these are the diseases which will transfer through heredity. So these can be prevented using rDNA technology. Understand the molecular event in biological process like growth, differentiation, and aging. So using this technology, the fields like growth, differentiation, and aging concepts have been improved, means we understood it in a very easy way ಅಂತ ಹೇಳ್ಬೋದು. So this is regarding the rDNA technology. So let us start with the solving of multiple choice question. Just check, can you able to see the screen? Just a minute. Yes. Can you able to see the screen?
Yes.
Okay. So the first question here is, I will be waiting in a chat box. Just a minute. Restriction endonuclease. Restriction endonuclease. What is the function? Option A, synthesize DNA. Option B, cuts the DNA molecule randomly. Option C, cuts the DNA molecule at specific sites. Option D, restricts the synthesis of DNA inside the molecules. Yes. Okay. Janvi said option D, Ananya C, Bhairesh C. Okay. So the right answer here is cuts the DNA molecule at specific sites, not at a random. ಎಲ್ಲಿ ಬೇಕಾದ್ರೂ ಅಲ್ಲಿ cut ಮಾಡಲ್ಲ. Specific site ಅಥವಾ specific nucleotide sequence ಅಲ್ಲಿ ಏನು ಮಾಡುತ್ತೆ cut ಮಾಡುತ್ತೆ ಅಂತ ಹೇಳ್ತಾರೆ. So option C is the right answer. Then gel electrophoresis is used for option A, construction of recombinant DNA by joining with cloning vectors, then isolation of DNA molecule, cutting of DNA into fragment, separation of DNA fragment according to their size. So what is the answer? Why it is used? Yes, I got 2 messages, 3. Okay. Bhairish, Ananya and Janvi, all of you said the right answer. That is option D is the right answer. To separate the DNA according to their size. Okay. Then third question. The linking of antibiotic resistance gene with the plasmid vector becomes possible with, I repeat, the linking of antibiotic resistance gene with the plasmid vector become possible with option A, DNA polymerase, option B, exonuclease, option C, DNA ligase, option D, endonuclease. Option C, C, C. Okay. So here cutting of DNA into fragments. Sorry, C, DNA ligase, linking of, okay. So the third option, the third answer is option C, that is DNA ligase. All of you given right answer. Then fourth question, polyethylene glycol method is used for option A, biodiesel reproduction, option B, seedless fruit production, option C, energy production from sewage, option D, gene transfer without a vector. Yes. ಪಾಲಿಇಥಿಲೀನ್ ಗ್ಲೈಕಾಲ್ ಮೆಥಡ್ ಈಸ್ ಯೂಸ್ಡ್ ಫಾರ್ ಆ ಮೆಥಡ್ ಅನ್ನ ಯಾಕೆ ಯೂಸ್ ಮಾಡ್ತೀವಿ ಅಂತ ಕೇಳಿದ್ದಾರೆ. ಜಾನ್ವಿ ಸೆಡ್ ಆಪ್ಷನ್ ಡಿ. ಜಾನ್ವಿ ಸೆಡ್ ಆಪ್ಷನ್ ಡಿ. Okay, so the right answer is option D, that is gene transfer without a vector. Gene transfer without a vector ಅಂದ್ರೆ gene transfer ಡೈರೆಕ್ಟ್ ಆಗಿ ಮಾಡಬೇಕು ಅಂತ ಬಂದಾಗ ಸೊ ಅಲ್ಲೇನಾಗತ್ತೆ, ವಿದೌಟ್ ಅ ವೆಕ್ಟರ್ ಮಾಡ್ಬೇಕಂದ್ರೆ ಪಾಲಿಇಥಿಲೀನ್ ಗ್ಲೈಕಾಲ್ ಮೆಥಡ್ ಅನ್ನ ಯೂಸ್ ಮಾಡ್ಬೇಕಾಗತ್ತೆ. Then fifth question, which one of the following is used as a vector for cloning genes into higher organism? Option A, baculovirus. Option B, Salmonella typhimurium. Option C, Rhizopus nigricans. Option D, retrovirus. Sorry for the disturbance. Option D, option D, option D. Okay. So, Rhizopus nigricans, fifth one. Fine. So here, this Rhizopus nigricans and the retrovirus. Okay. So many of you get confused regarding Rhizopus nigricans and the vectors because some of the characters will match. So here, even though, yeah, okay. So the right answer is option D, that is retrovirus. Sixth question. DNA or RNA segment tagged with the radioactive molecule is called vector, probe, clone, or plasmid. What it is called?
Option B, ma'am, probe.
Option D, probe. B, and also B. Okay. So the right answer is very easy and direct question. So the right answer is option B, probe. Good. Seventh question. Restriction endonucleases are enzymes which, read the options by yourself and tell me the answer. Seventh question.
Ma'am, option B.
Option B. Ananya said option B. What about others?
A, ma'am.
A. Option A. Janavi said option A.
Option A, ma'am.
Okay, good. So the right answer is option A, that is it make cuts at specific positions within a DNA molecule. See this seventh question and first questions. Here they asked the direct question. Here they asked adjust our enzymes with. The sentences or statements are quite different, but the question is same. Okay, read the question twice and then answer. Okay, now eighth question. Stirred tank bioreactors have been designed for addition of preservatives to the product. Option B, purification of the product. Option C, ensuring anaerobic condition in the culture vessel. Option D, availability of oxygen throughout the process. Stirred tank bioreactors ಯಾಕನ್ನ ಯೂಸ್ ಮಾಡ್ತೀವಿ ಅಂತ?
Ma'am, option D.
Option D, availability of oxygen throughout the process. Okay, any other answers?
Option D, ma'am.
Option D. Okay. Then what about Janavi?
Ma'am, B. Purification.
D is availability of oxygen throughout the process.
No, ma'am. Purification.
Purification, option B. Okay. So the eighth answer is option D, that is availability of oxygen throughout the process. That is why we will use stirred. So when you get the term like stirred, stirred means was stirring. So when you stir, there will be exchange of air very quickly. Okay. So that is why the substances will dissolve soon when you stir. ಹೌದಾ? ಈಗ ಏನಾದ್ರೂ ಕರಗಿಸ್ಬೇಕಂದ್ರೆ ಸ್ಪೂನ್ ಹಾಕಿ ನಾವು ಸ್ಟಿರ್ ಮಾಡ್ತೀವಿ, ಹೌದಾ? ಸೊ ಏನಾಗುತ್ತೆ, ಬೇಗ ಕರಗತ್ತೆ. ದ ಮೂವ್ಮೆಂಟ್ಸ್ ಆಫ್ ದ ಪಾರ್ಟಿಕಲ್ಸ್ ವಿಲ್ ಬಿ ಫಾಸ್ಟರ್. ಸೊ ಲೈಕ್ ದಟ್ ಸ್ಟಿರ್ರಿಂಗ್ ಅಂತ ಬಂದಾಗ ಅಲ್ಲಿ ಎಕ್ಸ್ಚೇಂಜ್ ಆಫ್ ಏರ್ ಆಗ್ಲೇಬೇಕು. ಸೊ ದ ರಿಲೇಟೆಡ್ ಒನ್ ಇಸ್ ಅವೈಲಬಿಲಿಟಿ ಆಫ್ ಆಕ್ಸಿಜನ್ ಥ್ರೂ ಔಟ್ ದ ಪ್ರೊಸೆಸ್. Ninth question, which of the following are used in gene cloning? Option A, nucleoids. Option B, lomasomes. Option C, mesosomes. Option D, plasmids.
Option D, ma'am. Plasmids.
Option D, ma'am.
Very quick answer. Good. Option D, plasmids. Good. Then 10th one. Yeah. In genetic engineering, a DNA segment or a gene of interest is transferred to the host cell through a vector. Consider the following 4 agents, that is 1, 2, 3, 4, in this regard and select the correct option about which one or more of these can be used as a vector. Means out of this, which can be used as a vector, you should say. 1 bacterium, 2 plasmids, 3 plasmodium, 4th bacteriophage. Sequence you should say.
Ma'am, option D.
Option D, 2nd and 4th. Okay. What about
Ma'am,
the
option A. Option B.
Option B, one only. Okay. Okay.
A, ma'am.
Option A. Good. So here the 10th answer is option D, that is 2nd and 4th. Plasmid and 4th one is bacteriophage. What they ask, consider the following 4 agents in this regard, that is genetic engineering, that is transferring to the host cell, and select the correct option about which one or more of these can be used as a vector. The option is, second one is a plasmid we can use, and the 4th one is a bacteriophage we can use. But this bacterium plasmodium is a protozoan which causes malaria. That is not related. Bacteria acts as a host cell. Okay. So here the correct answer is option D. I think Bhairesh answered it correctly. You said option D only, no?
Yes, ma'am. Ma'am.
Okay.
Ma'am.
Yeah, tell me.
Ma'am, why is RDNA technology called genetic engineering?
Yes, tell me.
Ma'am, why is called RDNA technology called genetic engineering?
Okay, genetic engineering. Why? Good question. So rDNA technology is called a genetic engineering. Engineering means what? What do you mean by engineering? What is the simple meaning of engineer according to you? I'm not forcing you. Just tell me, what do you mean by engineering according to your knowledge?
It is a professional work.
Professional work. Okay. Okay. So here, an engineer, what he will do means he will design certain things, including means by applying the knowledge basically of maths and science. Maths and science knowledge he will apply, and he will be professional in certain, like in computer science engineering, that is computer engineer will adapt his maths and science in the computers, mechanical engineer in the machines, biotechnology engineer in the biotechnology, that is in the living organism. So here genetic engineering means when we are learning rDNA technology, we come across the steps like isolation, that is separation, cutting, attaching, designing a new thing, designing a new DNA. So these all are the indirectly the methods which are involved in engineering, right? So here engineer, what he will do, he will adapt the knowledge of maths and science, and he will do a one certain or a one new thing or a one new machine. So here what we are doing, we are mixing up or combining the DNA and we are preparing a new DNA, a new format of a DNA to get the useful thing. So we are adapting the engineer qualities in the DNA or in a gene. So that is called as genetic engineering. Did you get?
Yes, ma'am.
Yeah. Okay. 11th question. Given below is a symbol of a portion of DNA strand giving the base sequence on the opposite strand. What is so special shown in it? Just see the DNA sequence and tell what is special about these DNA sequences. Option A, replication completed. Option B, deletion mutation. Option C, start codon at the 5' end. Option D, palindromic sequence of base pairs.
D, ma'am.
Option D, ma'am.
Option D.
Option D. Then 1 minute. Okay. Fine. Palindromic sequence, good. So when you see the sequence, you will get to know. Very easy question. But during exam, don't like ಏನ್ ಕ್ವಶನ್ ಅಪ್ಪ ಮುಂದೆ ಅಂತ ಅನ್ನೋ ಏನದು ಹೆದರಿಕೆ ಇರುತ್ತೆ. ಸೊ ಡ್ಯೂರಿಂಗ್ ದ ಎಕ್ಸಾಮ್ ರೀಡ್ ದ ಕ್ವಶನ್ ಟ್ವೈಸ್ ಅಂಡ್ ದೆನ್ ಆನ್ಸರ್. ಸಿಂಪಲ್ ಕ್ವಶನ್ಸ್ ಅನ್ನ ಬಿಟ್ಟು ಬರ್ತೀವಿ ನಾವು. Many times it happened with me also. Okay, question number 12. There is a restriction endomolecules called E. coli. What does co part in it stands for? The restriction endomolecule called E. coli anta ide. What does co ಅನ್ನೋ ಪಾರ್ಟ್ ಏನ್ ಅನ್ಸುತ್ತೆ ಅಂದ್ರೆ ವಾಟ್ ಇಸ್ ದ ಮೀನಿಂಗ್ ಆಫ್ ಇಟ್? ಆಪ್ಷನ್ ಎ ಕೋಲಾನ್, ಆಪ್ಷನ್ ಬಿ ಸೀಲೋಮ್, ಆಪ್ಷನ್ ಸಿ ಕೋಎನ್ಸೈಮ್, ಆಪ್ಷನ್ ಡಿ
Option D, coli.
Option
D,
D.
ma'am. Coli.
option D, choline. Okay. 12th question. Yeah. Option D, that is coli, is the right answer. Good. 13th question. Agarose extracted from seaweeds is used in option A, spectrophotometry, option B, tissue culture, option C, PCR, that is polymerase chain reaction, option D, gel electrophoresis.
Ma'am,
D,
option D.
ma'am. Gel electrophoresis.
All of you said option D, that is gel electrophoresis, is the right answer. Good. 14th question. Which one of the following technique made it possible to genetically engineer living organism? Option A, recombinant DNA technique. Option B, X-ray diffraction. Option C, heavier isotope labeling. Option D, hybridization.
Ma'am, option A.
Okay. What about others?
Ma'am, C.
Option C, heavier isotope labeling. Okay. Any other answers? Okay. So the right answer is option A, recombinant DNA technology. Fine. The given figure is the diagrammatic representation of the E. coli vector PBR322. Which one of the given option correctly identifies its certain components? Option A, ORI original restriction enzyme. Option B, ROP reduced osmotic pressure. Option C, HIND3 E. coli selectable markers. Option B, AMPAR antibiotic resistance gene. ಇದರಲ್ಲಿ ಒಂದು ಡೈಗ್ರಾಮ್ ಕೊಟ್ಟಿದ್ದಾರೆ. ಸೊ ದಟ್ ಈಸ್ ನಥಿಂಗ್ ಬಟ್ ಅ ವೆಕ್ಟರ್ ಡೈಗ್ರಾಮ್. ಸೊ ಅದ್ರಲ್ಲಿ ಸರ್ಟನ್ ಪಾರ್ಟ್ಸ್ ಇದೆ. ಯಾವ ಪಾರ್ಟ್ ಇನ್ ಹೆಸರು ಕರೆಕ್ಟ್ ಆಗಿ ಕೊಟ್ಟಿದ್ದಾರೆ. ಫಾರ್ ಒರೈ ಅಂತ ಹೇಳಿದ್ದಾರೆ. ಒರೈ ಅಂದ್ರೆ ಒರಿಜಿನಲ್ ರೆಸ್ಟ್ರಿಕ್ಷನ್ ಎನ್ಸೈಮಾ ಅಂತ ಕೇಳಿದ್ದಾರೆ. ROP ಅಂದ್ರೆ ರೆಡ್ಯೂಸ್ಡ್ ಆಸ್ಮೋಟಿಕ್ ಪ್ರೆಷರ್. ಸೊ ಈ 4 ಆಪ್ಷನ್ ಅಲ್ಲಿ ಯಾವುದು ಕರೆಕ್ಟ್ ಆಗಿ, ಯಾವ ಪಾರ್ಟ್ ಯಾವುದು ಕರೆಕ್ಟ್ ಆಗಿ ಲೇಬಲ್ ಮಾಡಿದ್ದಾರೆ. ಸೊ ದಟ್ ಯು ಶುಡ್ ಸೇ. 15th ಕ್ವಶನ್.
Ma'am, option D.
Okay. Okay. Option D is the right answer. That is AMPAR and TETR, which are called as antibiotic resistance gene. 16th question. PCR and restriction fragment length polymorphism are the methods for option A, study of enzyme. Option B, genetic transformations. Option C, DNA sequencing. Option D, genetic fingerprints.
Ma'am, option B. Ma'am, option D.
Option D. What about others? Janavi and Bhairesh.
D, ma'am.
Option D. Okay, genetic fingerprints, right answer. Both of you given right answer. 17th question. A single strand of nucleic acid tagged with a radioactive molecule. This is already repeated question, I guess. Option A, vector. Option B, selectable marker. Option C, plasmid. Option D, probe.
Probe, ma'am.
Good, very good. So the right answer is probe. 18th question. Which one of the following is a case of wrong matching? Option A, somatic hybridization, that is fusion of two diverse cells. Option B, vector DNA, that is site for tRNA synthesis. Option C, micropropagation, in vitro production of plants in large numbers. Option D, callus, that is unorganized mass of cells produced in tissue culture. Which is a wrong matching, they asked. 18th question.
C, ma'am.
Option A, somatic hybridization, fusion of two diverse cells. That is the right
No, ma'am.
statement.
C, ma'am.
C. In vitro production of plants in large numbers. No, it is the right answer. I mean, right statement actually.
Ma'am, D.
B.
Yes,
Okay,
ma'am, D.
Can you tell the statement?
Ma'am, callus, D.
Okay, D. So unorganized mass of cells produced in, no, that is a right statement. The answer is option B, that is vector DNA. What do you mean by vector DNA? The DNA of a vector or a bacteria which is carrying the plasmid. But they have given site for transfer, that is tRNA synthesis. That is a wrong statement. So the wrong statement is option B. 19th question. Which one is a true statements regarding DNA polymerase used in PCR, that is polymerase chain reaction? 19th one. Option A, it is used to ligate or stick introduced DNA in recipient cells. Option B, it serves as a selectable marker. Option B, it serves as a selectable marker. Option C, it is isolated from a virus. Option D, it remains active at high temperature.
Option D, ma'am.
D.
Option D, that is 19th one. It remains active at high temperature. Okay, good. Right answer. 20th question. For transformation, microparticles coated with DNA to be bombarded with gene gun for transformation. Okay, the microparticles which are coated with the DNA to be bombarded or inserted with a gene gun, which are made up of option A, silver or platinum, option B, platinum or zinc, option C, silicon or platinum, option D, gold or tungsten.
D, ma'am.
Gold or
Gold or
tungsten.
tungsten.
Okay, Ananya, what is your option?
Ma'am, D.
D, that is gold or tungsten. Fine. Then Bhairesh, what is your option?
Option D, ma'am.
Option D. Janavi?
D, ma'am.
Okay. So the 20th question is option D, that is gold or tungsten. So we completed 20 questions. We have more questions to be solved. Okay. So we will complete 5 more questions and then we will end the session. 21st question, biolistics, that is gene gun, is suitable for option A, disarming pathogen vector, option B, transformation of plant cell, option C, constructing recombinant DNA by joining with vector, option D, DNA fingerprinting.
Ma'am, option B.
B, ma'am.
Option B. Ananya said option B, that is transformation of plant cell. Yes, Janvi, what is your option?
B, ma'am.
Transformation of plant cell. Okay. Bhairesh?
Option B, ma'am.
Okay. So the right answer is option B, that is transformation of plant cell. 22nd question. In genetic engineering, the antibiotics are used for what? As selectable markers to select healthy vectors as a sequence from where replication starts to keep the culture free of infection. So what is the right answer? 22nd.
Option D, ma'am.
Option D, to
Yes, ma'am.
keep the culture free of infection. Remember the steps of RDNA technology. At last step, I said you why we will put antibiotics. If you put antibiotics, ಯಾವ ಬ್ಯಾಕ್ಟೀರಿಯಾದಲ್ಲಿ ನಮಗೆ ರೀಕಾಂಬಿನೆಂಟ್ DNA ಇಲ್ವೋ, ಸೊ ಆ ಬ್ಯಾಕ್ಟೀರಿಯಾ ಏನಾಗ್ತವೆ, ಅವು ನಾರ್ಮಲ್ ಬ್ಯಾಕ್ಟೀರಿಯಾ ಇರ್ತವೆ. ಸೊ ದೋಸ್ ಬ್ಯಾಕ್ಟೀರಿಯಾಸ್ ವಿಲ್ ಬಿ ಕಿಲ್ಡ್. ಯಾವ ಬ್ಯಾಕ್ಟೀರಿಯಾದಲ್ಲಿ ಪ್ಲಾಸ್ಮಿಡ್ ವಿತ್ RDNA ರೀಕಾಂಬಿನೆಂಟ್ DNA ಇದೆನೋ, ಫಾರಿನ್ DNA ಇದೆನೋ, ಆ ಬ್ಯಾಕ್ಟೀರಿಯಾ ಸರ್ವೈವ್ ಆಗ್ತವೆ. ಸೊ ಅಲ್ಲೇನಾಗತ್ತೆ, ಸೊ ದಿಸ್ ಆಂಟಿಬಯೋಟಿಕ್ ಈಸ್ ಕಾಲ್ಡ್ ಆಸ್ ಅ ಸೆಲೆಕ್ಟೇಬಲ್ ಮಾರ್ಕರ್. ಸೆಲೆಕ್ಟೇಬಲ್ ಮಾರ್ಕರ್ ಆಗಿ ಯೂಸ್ ಆಗತ್ತೆ. ಸೆಲೆಕ್ಟೇಬಲ್ ಅಂದ್ರೆ ಸೆಲೆಕ್ಟ್ ಮಾಡತ್ತೆ, ಏನನ್ನ, RDNA ಇರುವಂತಹ ಸೆಲ್ಸ್ ಅನ್ನ ಸೆಲೆಕ್ಟ್ ಮಾಡತ್ತೆ. ಮಾರ್ಕರ್ ಅಂದ್ರೆ ಏನ್ ಮಾಡತ್ತೆ, ಇಟ್ ಐಡೆಂಟಿಫೈಸ್, ಇಟ್ ಮಾರ್ಕ್ಸ್, ಇಟ್ ಶೋಸ್ ಅಸ್ ದ RDNA cells. Okay, so selectable markers ಅನ್ನೋದು ರೈಟ್ ಆನ್ಸರ್. ಆಪ್ಷನ್ ಎ ಇಸ್ ಅ ರೈಟ್ ಆನ್ಸರ್. ಓಕೆನಾ? 23rd question. Which one of the following represents a palindromic sequence in DNA? Read the options by yourself and let me know the answer.
A, ma'am.
Ma'am, option
A. Okay.
Option A, ma'am.
Okay, good. Option A is the right answer. 24th question. The colonies of recombinant DNA bacteria appear white in contrast to blue colonies of non-recombinant bacteria. Andre recombinant DNA ಇರೋ ಬ್ಯಾಕ್ಟೀರಿಯಾಸ್ ಏನ್ ಕಾಣ್ತವೆ, ವೈಟ್ ಕಲರ್ ಏನ್ ಕಾಣ್ತವೆ ಅಥವಾ ಲೈಟ್ ಕಲರ್ ಕಾಣ್ತವೆ. ನಾನ್ ರೀಕಾಂಬಿನೆಂಟ್ ಅಂದ್ರೆ ಯಾವ ಬ್ಯಾಕ್ಟೀರಿಯಾದಲ್ಲಿ RDNA ಇರೋದು ಇಲ್ವೋ ಅದು ಒಂಥರ ಬ್ಲೂ ತರ ಕಾಣಿಸುತ್ತೆ. ಯಾಕೆ ಅಂತ ಕೇಳಿದ್ದಾರೆ. ಆಪ್ಷನ್ ಎ, ಇನ್ಸರ್ಷನಲ್ ಇನ್ಆಕ್ಟಿವೇಶನ್ ಆಫ್ ಆಲ್ಫಾ ಗ್ಯಾಲಕ್ಟೋಸೈಡ್ಸ್ ಇನ್ ರೀಕಾಂಬಿನೆಂಟ್ ಬ್ಯಾಕ್ಟೀರಿಯಾ. ಆಪ್ಷನ್ ಬಿ, ಇನ್ಆಕ್ಟಿವೇಶನ್ ಆಫ್ ಗ್ಲೈಕೋಸೈಡ್ಸ್ ಎನ್ಸೈಮ್ ಇನ್ ರೀಕಾಂಬಿನೆಂಟ್ ಬ್ಯಾಕ್ಟೀರಿಯಾ. Option C, non-recombinant bacteria containing beta-galactosidase. Option D, insertional inactivation of alpha-galactosidase. So what is the answer for 24th question?
Option D, ma'am.
Option D, insertional inactivation of alpha-galactosidase. Okay.
Ma'am,
C, ma'am.
Option C is the right answer. Okay, 25th question. Which of the following is not correctly matched for the organism and its cell wall degrading? Degrading means breaking. Cell wall na break maro enzyme anta helidare. Option A, algae methylase. Option B, fungi chitinase. Option C, bacteria lysozyme. Option D, plant cells cellulase. Which is wrong, they asked. Which is wrong. Okay.
ma'am, option A.
A,
Okay.
ma'am.
Okay. So option A, that is algae methylase, is a right answer. Okay. Yeah. So today we completed the concept of rDNA technology and we solved 25 multiple choice questions on rDNA as well as biotechnology. So I want which concepts you want me to discuss tomorrow and which MCQs you want me to discuss. Yes.